Subscribe to our YouTube channel and press the bell icon to get the latest updates.
Justicia wynaadensis, a small branch shrub in Karnataka, locally known as Moddu Soppu, is used to make a sweet dish during the monsoon season by the inhabitants of the Kodagu district. Scientists have now identified two active compounds in the plant that may have anti-cancer properties. The team used a computer software to look at how different plant compounds interacted with an enzyme, thymidylate synthase. Thymidylate synthase is involved in making DNA for cell replication. As you may already be aware, uncontrolled cell replication is the underlying problem in cancer. The idea is that if this enzyme can be blocked, it will lead to DNA damage in the cancer cells and potentially halt cancer growth. So, a team from University of Bangalore and Siddhaganga Institute of Technology in Karnataka used computational biology to assess 12 chemicals extracted from the plant. Computational biology is a branch of science that attempts to understand and model biological processes using data analytics. The method helps speed up the process of scientific studies, allowing scientists to simulate the biological processes and study them virtually before investing time and money in laboratory experiments or clinical trials. Computational biology can help accelerate the process of drug discovery by identifying how a drug molecule will interact with a target enzyme. This method is being used by scientists worldwide to quickly scan through a library of thousands of molecules and pick out the ones that are most likely to work against a disease. For this, scientists need to know the 3D structure of the target enzyme. In this case, it is thymidylate synthase. Then they take the virtual models of the plant molecules and try to dock it with the 3D structure. Docking is an algorithmic process in which the molecules try to find the pockets in the 3D structure of the enzyme where they can bind with better binding energy. In physics, binding energy describes the energy that would be required to break apart a system of particles. The lower the binding energy, the less stable the bond between the two particles. The team in this study found that two compounds had greater binding ability to the enzyme than capecitabine, which is a chemotherapy medication. The first, campesterol, is a well-known plant chemical with a structure similar to cholesterol. The second, stigmasterol, is another well-known plant extract involved in structural integrity of plant cells. Of the two, campesterol is more stable and the team believes that it could represent a possible lead for further investigation and testing as an anti-cancer drug. Now, it is important to note here that the research is at a preliminary stage and further tests in the lab and in animals and in human models need to be conducted before one can claim that the compounds can indeed treat cancer. The research also does not mean that consuming the products made using this plant can cure cancer, as specific doses at which the plant extracts may work will also need to be ascertained. This is Mohana Basu, special correspondent at ThePrint. Follow us on social media for the latest updates.